So now we will be discussing the mechanism of concentration of the filtrate. As we have discussed in the early part of this chapter, that how it is important for the terrestrial animals to conserve water, and hence concentration of the filtrate, which finally becomes a urine, is very crucial. So let's understand how the concentration of the filtrate actually happens. So mammals have the ability to produce a concentrated urine, and to attain this concentrated urine we have two structures which are going to play a vital role. One is the Henle's loop, another is the vasa recta. Henle's loop, as you know, it is a part of your nephron, and vasa recta is the efferent arteriole which continues forward as the peritubular capillaries, and the peritubular capillaries which is just running beside the Henle's loop is the vasa recta. So the formation of concentrated urine occurs due to a excuse me, occurs due to a phenomena called as counter current. Okay, so what we are going to observe is that there is an opposite flow in the movement of the solution in the two limbs of Henle's loop as well as in the two limbs of the vasa recta. So the flow of filtrate in the two limbs of Henle's loop is in opposite direction and thus forms a counter current. The flow of blood through the two limbs of vasa recta is also in a counter current pattern. So if we have a look here in this diagram, you can see that this is the descending limb of Henle, right? And in this you can see the movement of filtrate is downward, whereas in the ascending limb the movement of filtrate is upward. So you can see the movement in both the limb is opposite, or we can say they are counter current. In a similar fashion, we can see the movement of the filtrate in case of the vasa recta. In the descending limb it is downward, and in the ascending limb it is upward. So this shows that there is this counter current mechanism occurring in both the two structure, which we have said will play a vital role in the concentration of urine. Another factor which is quite crucial for the formation of concentrated urine is the closeness that exists between the Henle's loop and the vasa recta. So the proximity between the Henle's loop and vasa recta as well as the counter current in them help in maintaining an increasing osmolarity towards the inner medullary interstitium. So for the formation of concentrated urine, it is must that we attain a increased osmolarity in the inner medullary interstitium. And how do we achieve that? It is through these two principles. One is the counter current mechanism, another is the proximity that exists between the Henle's loop and the vasa recta. So if we take the example of this diagram, you can observe that the osmolarity in the cortex region is very high, and as we gradually descend below, the concentration or the osmolarity increases. So from 300 milliosmoliter in the cortex to about 1200 milliosmoliter in the inner medulla, this gradient is mainly caused by sodium chloride as well as urea. So to increase the osmolarity in the inner medullary interstitium, we need the assistance of two salts. One is sodium chloride, another is the urea. It is the contribution of these two salts in the medullary interstitium which will increase the high percentage of osmolarity in the medullary interstitium. So let's have a look at how the sodium chloride actually contributes in increasing the osmolarity in the medullary interstitium. So sodium chloride is transported by the ascending limb of Henle's loop, which is exchanged with the descending limb of vasa recta, and finally sodium chloride is returned to the interstitium by the ascending portion of the vasa recta. So if you have a look here, this is the ascending limb of Henle's loop, and you can see that the ascending limb of Henle's loop is releasing sodium chloride and it is being taken up by the descending limb of the vasa recta, and finally this sodium chloride will be released into the medullary interstitium by the ascending limb of vasa recta. So in this way, the transport of sodium chloride from the ascending limb of Henle's loop, then getting exchanged in the descending limb of vasa recta, and finally getting returned to the interstitium by the ascending portion of vasa recta, helps in achieving a high osmolarity in the medullary interstitium. In a similar fashion, let's have a look how urea also helps in increasing the medullary interstitium. Similarly, small amounts of urea enter the thin segment of the ascending limb of Henle's loop, which is transported back to the interstitium by the collecting tubule. So if we have a look here, you can see that from the collecting duct, the urea enters into the into the ascending limb of Henle's loop, and it is returned back into the collecting duct so that it is released back into the medullary interstitium. So in this way, the medullary interstitium concentration or osmolarity increases. Due to this, what happens? The filtrate that is present in the collecting duct as well as in the tubules will be taken back into our system. So due to this, finally what is going to happen? We are going to observe that the urine that is formed is in a concentrated form. So the above described transport of substances facilitated by the special arrangement of Henle's loop and vasa recta is called the counter current mechanism. So you could see that there is a counter current movement of the substances in both the Henle's loop as well as the vasa recta. Right? So this mechanism helps to maintain a concentration gradient in the medullary interstitium. Presence of such interstitial gradient helps in an easy passage of water from the collecting tubule. Now, what was the need for maintaining a high osmolarity in the medullary interstitium? Because then only we could achieve our final target of, allowing the water from the collecting tubule and back into our system. So once the water leaves the collecting tubule, the urine that is formed is concentrated, and this is how we achieve a highly concentrated urine, and thereby we are making sure that we are retaining the water that was a part of the filtrate. So thereby concentrating the filtrate, that is a urine. Human kidneys can produce urine nearly 4 times concentrated than the initial filtrate formed. So